Before I start, I just want to say I'm thrilled to be part of the board, and I'm thrilled to be with you all here. It's a great honor. This has been an exceptional meeting as always. Such a privilege for us to be here. And in terms of shout-outs to those around the world who are watching, I'd like to give a shout-out to Alberta, Canada. I understand that we have a viewer from Canada also, so welcome. And the more the merrier as far as I'm concerned. So it's a pleasure to be able to talk to you, I think, with great optimism about the future of MSA research. This is an exceptional time, and we are poised, standing on the shoulders as we are of incredible research that's already been done in MSA and in other diseases to make a real difference to patients. In our own small way, where I work in Boston, we have a new facility that I think will hopefully capture our vision for MSA research. In this facility, we have state-of-the-art clinical and imaging facilities in the lower floors, and our research laboratories are in towers above those, bringing researchers together with clinicians, together with patients and their caregivers in one centralized location next to Harvard Medical School. And I really hope that initiatives like this can catch fire in facilities like this so that we can get the right teams together to solve very complicated problems like MSA. So MSA is a disease that can be thought of in multiple different ways, multiple levels. On the right-hand side, you're all familiar with these kinds of brain scans, in this case showing what we call pontocerebellar atrophy. Some people call this the hot cross bun sign. It reflects loss of myelin in a particular set of nerve fibers traveling down into the cerebellum in a patient with MSA-C. But we're going to focus at a different level mostly today, because it is probably at the cellular and subcellular level where we will unlock the mysteries about what causes this disease and what leads to its progressions, and therefore what we can do to slow that progression or prevent the disease, which is what we're all looking to do. And this here is a cell. I'm sure all of you are familiar with it. It's called the oligodendrocyte. It's a glial cell, a glue cell in the brain. And this particular cell is responsible for the myelination, the insulating cabling that goes around your nerve cell fibers that allows them to transmit at high speed. Well, one of the many pathologies, but the hallmark distinguishing pathology of multiple system atrophy is the damage that occurs to these cells. And so we're going to think a little bit about different levels, cellular and subcellular, and put those in the context of the research that's going on for MSA right now. We're going to talk about the tremendous advances, but we're also going to talk about the critical challenges that we face in 2017. And if there's an underlying theme to my talk, it is that we have to band together. This is a rare disease with very, very powerful consequences to individuals, and I believe powerful consequences for how we understand other much more common diseases. But in order to make a dent in it, we're going to need, as a research community, as a patient community, as a caregiver community, to come together. And we need to strategize about that. But we're going to focus at different levels, at the level of DNA, genes, proteins, cells, and ultimately how these can make up the body that is affected in this disease. And we're going to start with proteins, because if there's one area here where we've made the most advances, it is at that level. And so what are proteins? Most of you know this, but I'm going to review it anyway. Proteins are the structural and functional powerhouses of our body. They carry out function, they form structures, and it's an extraordinary amount of complexity that they capture in their structure. And in order to perform these exquisite functions, proteins need to fold in exquisite shapes. They are all uniquely folded, and they are geared, honed delicately to address their particular task in the cell. What one doesn't often appreciate with pretty diagrams like this of these proteins is that within the cellular context, they are incredibly crowded. Okay, this is a depiction of what it's like in our cell, and I don't think the video is working here, but which it doesn't really matter. But what I want you to capture here is the density at which these proteins are packed. Not only do they have to stay folded, but they have to move in that cellular environment, jostling around and retain their unique function. So this is an incredibly challenging task. And as soon as life decided to put a membrane around cells, the problem of protein folding and misfolding was born. Okay? So this is an ancient problem that all of cellular life has to deal with. Oh, there it is. There's the video of them jostling around. And so there's a very delicate balance of folding into these shapes and misfolding. And that balance is disrupted, particularly as we age, because the factors that we all have in our cells that protect against misfolding slowly become less efficacious as we age, and that creates this substantial problem. And as I mentioned, this problem is really as old as cellular life itself. So let's focus here on the right for a minute. This is a normal brain here, and this is a brain that is completely degenerated and ravaged. And in this patient, a single protein changed its shape and caused this devastation. This is the picture of a brain of a patient with Creutzfeldt-Jakob disease, a prion disease in which the soluble prion protein undergoes a switch and changes into an insoluble state. Now turn back the clock 1 billion years and take a look at these guys over here. These are yeast cells, the same cells that we use to brew beer and bake bread. Okay? They're streaked out on a plate, and you can see on the left are red yeast cells and white yeast cells. It turns out these yeast cells are genetically identical. They're the same cells genetically, yet one protein, just like in the prion disease over here, Creutzfeldt-Jakob disease, one protein has switched from a soluble state to an insoluble state, and you can see it's forming little green blobs here on the left, and that has caused a dramatic change of these yeast being red to white. Okay? So these are genetically identical, and yet a single change in shape of a single protein was capable of creating this change. And so prion diseases are really the archetypal disease here. Okay? And you've all heard of Creutzfeldt-Jakob disease in some way or another, because it's also known as mad cow's disease when it's infective, when it comes from us eating infected meat. And that single change in the conformation of the prion protein leads to a very aggressive rapid degeneration, which histologically, when you look at the brain microscopically, shows really holes in the brain where there used to be intact brain tissue. And it progresses very fast because a templating infective process takes over. One protein switches into an insoluble form. It meets another protein that's soluble and normal, but it templates. It infects that protein, and suddenly that normal protein is now misfolded and can transmit to the next protein, and that can spread from cell to cell. Okay? So this kind of process, this spectrum, is really what underlies all of these common degenerative, what we call proteinopathies, protein misfolding disorders. And so this entire family here, these are diseases you know: Alzheimer's disease, Parkinson's, dementia with Lewy body, frontal temporal dementia, prion diseases, ALS, Lou Gehrig's disease. These are diseases of protein misfolding. And for about 80 or 90 years since Alzheimer and Frederick Levy described these proteinaceous aggregates, no one knew whether they were what they were. No one knew whether they were causal, whether they were actually causing the problem, or they were just bystander events. And then two revolutions happened from the 1980s and then from the late 1990s. The first was these proteins began to be identified. We started realizing what they were. So the beta amyloid protein, the tau protein, the Huntington protein, these started being identified as proteins. And the second revolution was, of course, human genetics. And human genetics started to uncover families that had mutations in genes that led to very aggressive familial forms of these diseases. So mutations in alpha-synuclein could lead to Parkinson's disease early in life when a patient was in their 30s or in their 40s. And suddenly we realized that mutations in the gene encoding these proteins, like a mutation in alpha-synuclein leading to the aggregation of alpha-synuclein, suddenly we realized these are not just associated phenomena, they are causal. You can have a single change in a gene that encodes one of these proteins and you would get this disease early and aggressively early in your life. And so that was a really groundbreaking discovery because it allowed us to understand that these proteins are central to these diseases. And so what about MSA? So in MSA there is also the misfolding of a protein. And that had been known, and until the late 90s, it wasn't clear whether it was many diseases or one, and in some ways that's still not clear. As we simplify these diseases, we also realize they're more complex than we thought. But there was really a landmark discovery in the late 90s, and that was the identity of that protein that creates the hallmark pathology of MSA, unifying Shai-Drager and the other members of the family was discovered. And that identification was, of course, as you well know, that the protein alpha-synuclein was not only misfolding in Parkinson's disease and dementia and Lewy bodies, but was also responsible for the misfolding that was characteristic in those glue cells, the oligodendrocytes, of multiple system atrophy. And so that was really a critical advance and a key turning point, and it still is, looked back on as really a magical moment in MSA research where suddenly there was a culprit, there was something to go by, and there was a science, an emerging science that had been built around Parkinson's disease that could now be applied to this rarer disease. And while we're on the subject of proteins, about 15, 17 years later, another key advance at the level of proteins came from Stan Prusner's group. Now, Stan had won the Nobel Prize for identifying, and naming infectious proteins, prions, years ago. And he also started to show us that the protein that was aggregating in synuclein had different properties to the protein that was aggregating in Parkinson's disease and dementia with Lewy bodies. The problem with using the word prion, there is a problem, is that it implies infectivity, and we can have a discussion about that. But what I want to focus on here is not so much how related these two disease groups are, but the fact is that there is something unique about the property that correlates with the aggressiveness of the disease. Again, making us feel that unlocking the mysteries of alpha-synuclein and what it does to cells in the brain may be a critical aspect of understanding what multiple system atrophy does. And of course, therapeutically, that opens up options too. So this is a very complicated diagram. And generally when you see complicated diagrams, it means we don't understand exactly what's going on. And I don't want you to get boggled with the complexity, but there's a cell up the top and a cell down the bottom. And each of these different blobs in different colors are proteins that are misfolding. And what each of these little pathways shows, again, you don't need to read the text, is that there are many, many mechanistic possibilities for how one protein spreads from one cell to another. And we think such a spread occurs for the alpha-synuclein protein that's misfolding in MSA. And why is that important to you? Well, of course, it opens up ways of thinking about therapies. Because if we could block these forms of release, or if we could block these forms of uptake, or the misfolding process itself, which is called seeding, well, we might have therapeutic strategies. And so this was a great advance because it allows us to think about ways to block seeding, to block transfer, and to soak up synuclein, for example, with antibody therapies. Immunotherapies, as you know, are revolutionizing cancer treatment. Well, we might be able to unleash them to revolutionize the treatment of neurodegeneration as well. But before we jump to conclusions and get too optimistic about these types of treatments, we have to understand how little we know. The reason this is, as I said, is so complicated is there are so many possibilities right now, different experimental systems, conflicting data, and none of this will add up to a coherent story or a therapy unless the basic biology is worked out more concretely. Okay, so what does the connection of Parkinson's mean for MSA? Just a few points. Well, of course, the MSA community can now benefit from 20 years to more than nearly 30 years of Parkinson's research into alpha-synuclein. This can be at the level of biology, biomarkers, treatment therapies, stem cell technologies. We've built the science, for example, in our lab of Parkinson's disease stem cell biology. We can now use those insights for MSA. And the Parkinson's disease field has strength in numbers that the MSA field does not have. This is a gap where we have to, again, team together to build this on this work and bridge to advances in MSA. But what the strength in numbers for Parkinson's disease means is that we can look at large populations of Parkinson's disease patients and make conclusions. And some of those may have very, very real consequences for MSA. So as an example, I'll give you this example of a paper we recently contributed to. It was published just a few weeks ago, and Clemens Scherzer in my department drove the study. And what Clemens found was that, and I'm sorry the slides aren't fully coming up here the way it's projecting, but what he found was what's actually written there is beta-2 adrenoceptor agonists have consequences for alleviating Parkinson's disease risk. And what are these beta-2 adrenoceptor agonists? Well, they're medications like salbutamol that we puff for asthma. Okay, and they lower the levels of alpha-synuclein in cells. And remarkably, Clemens showed, and again, apologies that this is not all coming out on this screen here, but what Clemens showed was that if you took now thousands of patients, and these were well followed in Norway, and if you looked at those patients or those people who had been taking, this top line here is salbutamol, who had asthma, who were taking this medication, they had a much lower risk of developing Parkinson's disease over time than the remainder of the population. And so a commonly used FDA-approved drug has the chance of lowering the levels of alpha-synuclein. And actually, when you have strength in numbers like you do for Parkinson's disease, you can actually make causal inferences about risk at the population level decreasing with patients who are taking those medications. Now, am I telling you to start going and puffing salbutamol? Absolutely not. Okay? This is an untested hypothesis, but it is an extremely exciting one. It needs to be tested rigorously in Parkinson's disease and in MSA, and appropriate proper trials need to be established to figure out if it's effective and if it's safe. But it's promising, and it's one way in which the MSA community can benefit from the Parkinson's research that is ongoing. Then, of course, the development of animal and cellular models. These have been made for Parkinson's disease, and now we are applying those same kinds of principles to MSA. Mouse models of MSA were rapidly developed and characterized, and of course MSA itself offers a tremendous opportunity for the Parkinson's disease field. Unfortunately, we know that MSA progresses more quickly than Parkinson's disease, but this means Parkinson's researchers have become energized to test out therapies targeting alpha-synuclein in MSA patients. And so this symbiosis between the fields, I think, is going to be critically important. And of course, MSA classifies itself as a rare disease, and that allows for certain regulatory flexibility in getting treatments to MSA patients, and the Parkinson's research community is intensely interested in that. So we've covered these. And a great example are the emerging, as I said, immunotherapies, antibodies directed at alpha-synuclein that I think are very exciting to be tried in both patient populations. So we've focused a lot on this protein alpha-synuclein, but I do want to bring it back to genes. Remember what I told you, that we knew a lot about the proteins in the 1980s and 90s that were clogging up brain cells in Parkinson's and Alzheimer's, but it was critically that relationship to genetics that built a bridge to the science that has really driven the explosive discoveries in those more common diseases. We need such a bridge for MSA because causal connections are tied to our genes. Okay? And so every time we try and model something, or we try and figure out if a disease is one disease or many diseases, or we try and match a therapy to a particular patient, we need to understand the genetic basis of that. Even if we're interested in environmental factors that can cause a disease, we need to understand how those environmental factors interplay or interface with our genetic vulnerabilities. Okay? So there is a great amount of importance in basic research in genetics. And how do we find genes? I just wanted to survey this field very simply for you so that you had a framework when you go to your physicians and certain terminology is potentially thrown at you. Well, we're looking for particular mutations, and in the old days, and by the old days I mean the late 20th century, the way to find a gene was to take a family where there was a trait, something that was tracking from generation to generation, and painstakingly, often over 10 to 20 years, try and map which part of the genome was tracking with the condition, and slowly get finer grained knowledge until you narrowed it down to the particular gene that was mutated. So that was called linkage mapping. More recently, you might have heard the term GWAS, Genome-Wide Association Studies. So these are studies where you compare affected individuals from unaffected individuals, and what you're looking for here are common variation in genes. So these are common in the population that slightly increase your risk of getting a particular disease. Okay? So these are called common variants, and you might hear the term SNPs, stands for single nucleotide polymorphism. It's a big phrase, but all it really means are markers on the genome that codify what a particular address in your genome looks like. And if there are some addresses that correlate with disease a little more than others, and they do so with high statistical significance, they're called positive hits in a GWAS study. And finally, really revolutionizing genetics is our ability to not just look at markers like SNPs, which are like little post boxes along the genome, but to actually sequence the entire genome, or the part of the genome that codes for proteins, which is called the exome. We can sequence that now at much, much lower cost. So you can sequence a genome now for $1,000. And so that's what we call whole genome or exome sequencing, and that's looking for rare variants. And so there have been some great advances in the field of MSA genetics, and there have also been challenges. So the first multi-generation families that have been described for MSA, which is generally considered a disease that does not transmit in families, but in certain multi-generation families in Japan, there was a finding of variation or mutations in this particular gene that encodes a part of a pathway that ends up making something called coenzyme Q, which many of you may take as a supplement. And there are mutations in that correlated with the MSA trait in a particular family. But there were controversies in that. And was that finding true, or was it not? It most likely relates to different genetic backgrounds. Variation that happens in family is rare, and our populations have diverged so much over the last 10,000 years that it is becoming difficult to use just genetics itself to validate or confirm these findings. And so a lot of work needs to be done, but this was a very important advance for the field. And many of you know, I've been asked questions about it already, that there have been studies, genome-wide association studies like this one, that have failed to verify or failed to identify any gene that rises above the noise for multiple system atrophy, whereas they have been successful for Parkinson's disease. And so there are ways in which we have to work together and more smartly to interrogate our genetic information to find those genes in MSA. It's a harder problem because it's a rarer disease. But the Parkinson's field gives us a glimpse on what is going to be possible. And in Parkinson's disease, again, you don't need to read these gene names, but we now have a number of genes where we know rare genes where we know mutations lead to Parkinsonism and in many cases to actual Parkinson's disease that arises from the misfolding of alpha-synuclein. And again, you don't need to read these. This is just to say that those genetic genome-wide association studies down here and those exome genome sequencing studies up here are yielding high dividends for Parkinson's disease, and in this list is alpha-synuclein itself, both a hit in the common genome-wide association study and a hit in these rare variant studies. So alpha-synuclein is not only aggregating in Parkinson's, but as I mentioned before, it is definitively linked. Patients with variation in that gene are more likely to get the disease. Patients who have mutations in that gene are likely to get that disease earlier and in a more aggressive form. And this is what's called a Manhattan plot because it looks like the skyline of Manhattan. Again, you don't need to know the names of these genes. This is a paper just published a couple of weeks ago in the field of Parkinson's genetics in the journal Nature Genetics, which has greatly increased the number of genetic factors that we now know lead to an increased risk of Parkinson's disease. And in our own group, that finding by geneticists, as incomplete as it is, as fantastic as this is, I should say that probably for Parkinson's disease we still know no more than 10% of the hereditary factors that contribute to the disease, and that's after around 20 years of work. So even in the Parkinson's field there is much to be done. Nevertheless, these significant advances have allowed us to do something very important, and that is to ask how these many different genes are connected to that protein alpha-synuclein that misfolds in the brains of Parkinson's and MSA patients. And so recently we carried out a large study to do this systematically, and I'm not going to go through the details of this study, but just to tell you that we have a variety of models ranging from simple cells like yeast cells that I mentioned before to complicated brain cells that we make from our patients with Parkinson's disease to start to understand how this misfolding problem relates to the many genetic factors that were coming out of Parkinson's genetic studies. And this was funded in part by the MSA Coalition, so I am tremendously grateful to them for allowing us to finally be able to view at a subcellular level how alpha-synuclein is perturbing our cell biology. Okay, so we've created the first map which shows us how the pieces of the puzzle fit together. Okay, once again, I don't want you to delve into the complexity of this map. I can answer any questions about it, but what this is really a map. Every circle here, and some are just bigger than others to emphasize them, but every circle is a particular gene or protein, and every color just reflects a different process in the cell. And for the first time we can visualize the different ways in which alpha-synuclein is affecting different processes in the cell and how they are interconnected with that protein. You might notice little targets that are here. They are the first examples of components of the map where we have developed small molecules, in one case an FDA-approved molecule I'll talk about later called FK506, that can target genes or proteins in that map. And what is this, taking a step back, what does this allow us to do? Well, one day here's the vision, that we'll be able to create a cellular map for every patient, and be able to understand how the genetic makeup of a patient is related to targets that can be ameliorated by particular small molecules or gene therapies, so we can match them to an individual patient's risk. Okay? So this is the first glimpse we have of how to put puzzles like this together. And so as I said here, just to summarize, this is how alpha-synuclein, which sits here on the map, is connected to many disease-related proteins involved in many distinct cellular processes marked with these colors. We have defined relationships to druggable targets where we know small molecules are engaging the machinery, so we can relate specific genes to specific small molecules. This map, understanding how this general map works, allows us to understand vulnerability of different cell types to alpha-synuclein toxicity. So how, for example, does a dopamine neuron differ from an oligodendrocyte in the way it is responding to alpha-synuclein? Well, we now have at least a guide of what to look for in these cells to start making these distinctions. And finally, this map includes many known genetic risk factors for Parkinson's disease. So could there be in this map new genes related to Parkinson's or MSA, deeply related to alpha-synuclein biology, that are going to be driving our risk for getting these diseases? And so with this map in hand, we have started to look at that. We've done what's called targeted exome sequencing, taking 500, 1,000 patients, and started to sequence these genes that we think are important, looking for genetic variation in patients. And it's starting to yield dividends. This is a new version of the Manhattan plot we're just starting to produce, and what you can see here are two well-known Parkinson's genetic risk factors are coming out of our studies, but each dot on this plot is another potential genetic risk factor for the disease, but we need to do a lot more work to make these findings solid. Will this work for MSA? Well, we think so. We've taken the first steps with Dr. Tsuji in Japan, who I mentioned a few minutes ago, and we have created the first plots that show very interesting genes rising above the noise in his cohort. And I should say one in particular that was of interest to us was this particular gene or protein NED4. We had published a paper several years ago showing that we could create a small molecule that could drug that particular protein. So wouldn't it be great if we could identify which patients had variation in their genes that related to this protein, because maybe there's a molecule or a group of molecules that could target the disease-causing process in that patient. But the bar is high. This is nowhere near a done deal. This is intriguing preliminary data that makes us feel that we're on the right track, but so much needs to be done. And in order to do this, it's not just geneticists or clinicians, but we have to work with cell biologists to have the model systems in which we can study this in the lab. So a little explanation for why basic research in genes needs to come together with basic research in cellular models. Why are genes so important? Well, in the end, we need to know how many diseases we are dealing with here. We need to know how to stratify patients for a clinical trial, not only to know that we're appropriately putting treatments, matching treatments to particular genes, but also to know that we're actually comparing apples with apples, and we're not just mixing up patients who have different genetic factors, because we'll lose the data in the noise. We also need to understand the outcomes of clinical trials. Right now, we take a cohort of patients across multiple centers, we expose them to a particular drug, and we say, well, did the treatment succeed or did it fail across the entire cohort of patients? And we run a statistic, and if it says, well, it failed, that's the end of the trial, and often that kills the research into that pathway or that molecule. But what if I told you we could go back into that patient population and figure out which combinations of genes look to be enriched in those patients who actually responded or looked like they were responding to a therapy? You would then get so much more information from that trial. And so that's why we need this genetic information. And why do we need cells? Well, in the end, we need cells in order to test potential treatments. We need to be able to experimentally design the appropriate therapies and refine them in the lab, so we need cellular models. And if we think that the environment plays a big role in MSA and Parkinson's disease, we need a model system that is not as tedious and as cumbersome as a mouse model. We need a faithful model system to understand how genetic factors interact with environmental factors. Okay? And that brings us to the final part, and that's really looking at cells themselves and bodies, by which I mean in this context animal models. Okay? So that discovery in the late 90s of synuclein being involved in MSA really empowered the field to make models of the disease. And so in another key advance, 2002, we started seeing the first mouse models made, a tremendous advance in which alpha-synuclein was being overexpressed in those glue cells, the oligodendrocytes, and driving neurodegeneration. So this was an exciting moment where it looked like there was a model of the disease. And it's important to note, though, that there were big assumptions engineered into these models. So both oligodendrocytes, these glue cells, are absolutely where the hallmark pathology occurs in this disease, but let's not forget the neurons, the brain cells. They are also affected in this disease in a dramatic way. So which is affected first? It's a chicken or egg problem, and we have not resolved it. And part of the reason we haven't resolved it is we haven't found critical genetic factors that we can anchor our models on. And so it's really important for you to know that those first mouse models were exciting, but they were also driven by a bias. And that was that if you force the protein to go up in levels in oligodendrocytes, those glue cells, that's the driving force in MSA. We haven't figured that out as a field yet. It was the right thing to do, but it is not a done deal. Okay? And so it may not come as a surprise that key molecules that worked in those mouse models did not work in humans. There are many possible reasons for that. The models may be fine. Maybe it was the way in which the clinical trial was designed. There are many, many things that need to be worked out. But I just want to caution you that even though there are these models that are exciting, there is much that yet needs to be done at the basic science level for us to understand this disease. And yet, what came out of this amazing tour de force work for clinical trials in MSA? Well, some really, really great things came out of it. Let us not forget that out of the incredible hard work of patients, clinicians, and scientists, we now have the science behind how to actually undertake a clinical trial in MSA. And this is thanks to amazing work from many people that you're familiar with, and I'll highlight just a few of them. The first was the group of Philip Lowe and Wolf Singer at the Mayo Clinic, who have started to publish papers to actually let the research community know that if you want to design a clinical trial for MSA, this is how you do it in a way that you're actually powered to generate results. And I find this work fantastic. So by building the science, they were able to take work that had been going on in Korea for mesenchymal stem cells, and they were able to, as you know, start to do the first clinical trials for mesenchymal stem cells because they had the framework, the clinical trial framework set up to do it. So I think that's a tremendous advance. And then there are other groups, and you've heard from Lucy and Horacio Kaufman. You've heard in the past from Jeremy Schmarman. These groups have been doing fantastic work on the natural history of these diseases, because if we don't understand how this disease progresses, we're not going to be able to design a clinical trial or understand the basic biology and heterogeneity of these diseases. So these are setbacks for sure when a clinical trial doesn't work, but they're also key advances, and I really want to drive that home, that time has not been lost in all of that brilliant work that was done in the natural history studies and in the clinical trials. And so, of course, there are active, people here have heard of EGCG, the key ingredient of green tea that's being tried. AstraZeneca has a myeloperoxidase inhibitor that targets inflammation that is being clinically tried for MSA. There's interest in a drug called nilotinib. I've talked about synuclein lowering therapies or antibody therapies. Well, all of this work gives us the framework in which these treatments can now be tried, which is a tremendously positive thing for the field. But a key challenge is going to need to be to create better cellular models for this disease, and I want to drive that home today. And so there was an amazing advance, and again I've talked about it here and in other places that you might have heard about this advance. Many of you are familiar with it, that was made by Shinya Yamanaka, now a Nobel laureate from Japan, who showed us that we could take adult cells, so skin cells, hair cells, even living cells that get flushed out in your urine, could be turned into human embryonic stem cell-like cells called induced pluripotent stem cells. And this was a game changer for modern medicine, and it allows us to take a small piece of skin from our patients or a blood sample, turn them into a cell that we can now convert into a brain cell and even little mini brains, for example. So in our lab right now, we make little mini brains from our patients that fold like a fetal brain would fold. We make retinas, so all of you know that the window into our brain are the eyes, and there's a neural part of the eye at the back of the eye called the retina. I have an amazing postdoc who is generating layered retina in the dish so that we can study diseases that affect the retina in a dish. So we are making advances based on this seminal work by Yamanaka and colleagues to study diseases from our patients. And so how this basically works, as I mentioned, is, for example, a skin biopsy or a blood sample. These are skin cells, the fibroblasts that we grow. We reprogram them through insights from Yamanaka's group into these colonies of thousands of cells. Each of these cells can become any cell in your body, and we pattern them into different cell types, in this case brain cells or neurons. These cells can be propagated indefinitely in the lab, differentiated into neurons or those oligodendrocyte cells we've been talking about. And here's something really important for the MSA field. We might not have the gene, but what we know is that when we take an iPS cell from a patient, we've captured that patient's genome. So therefore, we know that we've captured the genetic code of a patient who is susceptible to the disease. So even though we don't know what that gene is, we know we've captured it in the dish, okay? It doesn't make it an easy task because it's possibly many genes and we don't know what we're looking for exactly, but it really is a powerful aspect of this technology. And of course, and critically important for us pushing this field forward, the results from these iPS cells need to be compared to patient brain. And here's a shout out for my amazing patients who over the last decade have donated their brains to me. And while that's sometimes a morbid thing to bring up, but those generous people have enabled our research and our validation of this entire technology. And I've talked to you about how we've used those donations in the context of Parkinson's disease before. We need to now move the field forward for MSA. So for Parkinson's, we've established proof of principle. I've talked about it before. I'm not going to talk about it in great detail, but a few years ago, we published that we could put the protein alpha-synuclein into those yeast cells. Remember I talked about those yeast cells that we bake and we brew with? Well, it turns out that they die under the stress of alpha-synuclein just like our brain cells do. And what we can do to yeast cells is we can chop and change every gene in their genetic code and screen them against millions of molecules. And so we did that for the synuclein protein. We identified targets, molecules, and we could reverse the defects in brain cells that we generated from Parkinson's patients. And in fact, the findings that we made in the dish allowed us to find new things from the brains of the patients who have those diseases. So in this case, we had yeast cells, we had brain cells from living patients, and we had family members who had donated their brain so that we could verify and push the field forward on the basis of this tripartite, three-part model system. One thing we could do with Parkinson's disease, though, that we cannot yet do for MSA, and I'll drive it home again, is in Parkinson's disease cells, we can use a cutting-edge technology known as genome editing to correct the mutation. So how did we start this work for Parkinson's disease? We generated stem cells from patients who had a mutation in alpha-synuclein, and we used genome editing to genetically correct it in the dish so we knew we were comparing apples with apples. A patient's cell was being compared to her own cells with that single gene mutation corrected back. So we knew that we were dealing exclusively with the consequence of that one mutation. And so that was a powerful way in which we could begin this process because we knew more about the genetics. And so now, can the same approach be applied for MSA? Absolutely. Is it much more difficult? Absolutely. And we have, as I said, these amazing patients who have donated tissue, brain tissue, from which we have now generated living skin cells. Then these, you can see these skin cells are turning into colonies here of stem cells, and here you can see a colony of stem cells growing from this patient, which we can propagate indefinitely in the lab, generate all of the different cell types of the brain in which we can now study the disease process. And here's an example of us using insights into the protein, this alpha-synuclein, this specific form of alpha-synuclein that's aggregating in multiple system atrophy. This is a patient's cells. They're made into brain cells, and they are being exposed to misfolded alpha-synuclein protein. And what you can see along the nerve fibers is you can see these bright spots, and this is where the alpha-synuclein is starting to aggregate in these patient cells. We're hoping with this technology to create those kinds of cellular models that we need. But what are the challenges? There's no gene for MSA, so we can't correct it in the dish. How many lines, in other words, how many patient stem cell lines are we going to need? This is incredibly expensive, resource-heavy work. How many are we going to need to establish the efficacy of, for example, a potential therapy? How many different diseases are we dealing with? These are challenges we'll need to face. How many cell types do we need to generate? And are critical environmental factors missing? We need to understand what those factors are. But thanks to the MSA Coalition and other people who have generously funded the work in our lab and many labs around the world, this technology is now growing and available and is giving us a great source of optimism for finally having models that capture the potential complexity. And so these are the same questions rephrased in a slightly different way. And so, in a nutshell, we need to create stem cell banks for this disease. We need to use them to identify molecular defects in this disease, for the discovery of biomarkers, and for our ability to screen drugs and one day match them to individual patients. And I'll give you one example of something we're moving forward into the clinic now, and this is the immunosuppressant FK506. At high doses, it suppresses the immune system, but at low doses, it has very potent effects on the toxicity related to the protein alpha-synuclein. We are now at the point of wanting to know, can we establish biomarkers in the clinic that can establish what we call target engagement, that in a living patient with MSA or Parkinson's disease, the drug is hitting an appropriate target that we know it was hitting in one of our animal or cellular models. And if we can do that, can we design a well-thought-out therapeutic clinical trial? And so here's something a little bit about tacrolimus, or FK506, discovered in the 80s, FDA approved in 1994 for immunosuppression, now used in patients who get multiple types of transplant. It readily crosses the blood-brain barrier. We know a lot about how it gains access to the brain. And more importantly, it reverses the toxicity not only in yeast cells, but all the way into living rats and into neurons that have been derived from Parkinson's disease patients. And we have, and this is not my work, but it was one of my colleagues who was at MIT MIT, but now runs her own lab at Northwestern, Gabriela Caraveo. She identified the mechanism through which it's protecting against alpha-synuclein toxicity, so we have a way to move forward with it in a logical way into the clinic. So you don't want to run before you walk with this. You want to understand the biology from the grassroots up, but once you do it, you go in with full vision into the clinic. So one thing that Gabriela noticed, and here she is here, is that neuroinflammation was reversed in mouse models that were carrying the toxicity of alpha-synuclein. And so in my department, we teamed up with Tarun Singhal here, who had been working with a new PET ligand, so a new marker which can be imaged with PET scans that can monitor exactly the type of inflammation that was getting reversed in the mice. And so now we want to ask the question in our patients, can we visualize changes in this marker in the course of 1 year in patients who have MSA? And so fortunately the Harvard Neurodiscovery Center has funded us to do this, and I'm so excited to be able to show you the first images from just a couple of weeks ago of one of our patients who has undergone such a scan, and indeed we are starting to see signals in the critical areas affected in MSA of this marker being lit up. And we want to be able to see now if it progresses over a year. If it does, we hope that this gives us an avenue to a well-thought-out clinical trial in which we can monitor target engagement of the molecule of interest. And so finally, I leave you with the message that I think is throughout the whole talk, and that is that a team effort will be the key to understanding and treating MSA, I believe. This teamwork is going to center around patients and is going to need to empower patients. If you're going to give your samples to us, you deserve to know what happens to those samples. You deserve to have those samples shared and being used in an effective way. And as the coalition, our responsibility is to figure out a way in which to make this happen. And how can we get all the key stakeholders, together, to work together, and in the scientific discipline, not just neurologists, not just geneticists, but epidemiologists who can tell us about environmental factors, molecular biologists, cell biologists, people who understand computational biology, so we can interrogate those small genetic sets from MSA patients in the smartest way possible to get the most bang for our buck. Okay? And we're going to need to crowdsource this. We're going to, in order to empower you, we're going to need to use the full weight of awesome technologies that we now have to gain access to genomes and samples. And it really has to be a process that empowers you, the patients, and you, the caregivers. We absolutely should not reinvent the wheel. We should be looking for places that have already done this successfully with other diseases. You've heard in this series of the GLOMSAR initiative, the amazing initiative spearheaded by the Kaufmans, which should absolutely be at the center of such an initiative. I could say in our own cohort of hospitals, Clemens Scherzer and others are driving the Harvard biomarker study, where they follow nearly 3,000 patients now with Parkinson's and Alzheimer's disease. We should be able to use that framework for MSA. So finally, some acknowledgements. These are the people who have played absolutely pivotal roles to the science that I've presented here today. Among them are mathematicians, cell biologists, yeast geneticists, systems biologists, all coming together to try and solve these problems. And here's an amazing woman, Sue Lindquist, my mentor at MIT, who passed away last year. So a call out to her. She was a person who could really bring people together, and we need people like Sue to catalyze these amazing collaborations that can really change the world. And finally, a thank you, a special shout out to the coalition, to my colleagues there now, and to the patients and the caregivers. Sometimes you come up to me and you shock me and you thank me for what we do for MSA, but it's we who should be thanking you, okay? You enrich our lives and you make us better people. So thank you so much. I leave you with the last glimpse of one of my fabulous patients who sent this photo of herself climbing a mountain the other day. Each of you in your own way climbs mountains every day. Thank you for bringing us closer to a cure. Thank you so much.
Thank you, Dr. Khurana. Okay, I'm going to ask at this time that Dr. Horatio Kaufman, Dr. Lucy Northrup-Kaufman, Dr. Amanda Peltier, and Pam Bauer of our research committee, chair of our research committee, come up to the seats up front for Q&A. We have about 25 minutes for your questions. Go ahead and write them on the cards. Again, our volunteers will be coming around and picking them up, and we'll do our best to get through as many of them as we can. Come on up over here and have a seat, please. Let's get... Everybody this morning was sitting in the front. I think so, yes, thank you. And could we get the other wireless speaker up, mic up here? Thank you. I'm going to pass it back to you, and you guys can pass things.
Okay. There are two microphones. There's one on that side too. Okay, good. Maybe I'll get one more chair.
Dr. Biagiani, would you care to join the panel? We would love to have you. Wonderful. Thank you.
All right. One question here is about alpha-synuclein. This question comes from online. Does it cause the brain to misfold faster than PD? So I interpret that question to mean, does the protein misfold more quickly and more potently in this disease, MSA, than it does in Parkinson's disease or dementia with Lewy bodies? To the best of our knowledge, yes, it does. Okay, the part of the reason this disease may be more aggressive is that the protein may misfold more potently and may be more transmissible to accompanying or associated proteins that are in the vicinity. And I'm phrasing this carefully, and it sounds like a lot of jargon, because when I say transmissible, I don't necessarily want you to think of infectivity. I think that's very much an open question in the field, and it's one of great concern to patients that, if people are attaching the word prion, which has been attached to mad cow's disease, to MSA, does that mean this is an infectious disease? At this point, there's absolutely no evidence for that, okay? But when we think about transmissibility, that is within a cell and also from cell to cell, and we think at least under the artificial experimental conditions that have been tested right now, yes, this is transmissible, and yes, there is something more transmissible about the MSA-related synuclein. But there is much work to be done, and I think we need to be aware of how limited those early studies are. Okay, I'm just looking at the next question. So this question, I suppose I'll take as well. A second question from online. If PD-MSA is linked to the buildup of alpha-synuclein, why is MSA more rapidly progressing? Is it dependent on the part of the brain being damaged by the protein? So these are great questions. Yes, the protein is fundamentally the same. It's the same protein, but the way in which it folds, which scientifically we know as the strain of the protein, may be different. Remember I showed you at the beginning those yeast cells that were identical genetically? Well, and they only differed by the folding of a protein. Well, those different folds can be called strains. And we think that MSA synuclein is a different strain of protein than the Parkinson's disease or the dementia with Lewy body strain of synuclein, and it's more potent in causing problems for cells. And as the question implies, there are different cells affected. So the fact that you are affecting oligodendrocytes may have great implications for MSA, not just the parts of the brain, but the actual cells that are affected. Because remember, an oligodendrocyte, a glue cell, connects with multiple brain cells or neurons. So if one gets affected, it's quite possible the disease is more aggressive because many more cells will then go down because they're being contacted by a single glue cell, oligodendrocyte, that's been affected.
I'll take this one and then we can move it down. So the first question is, how do you volunteer for basic research clinical trials? So probably the easiest thing is if you go on all clinical trials in the United States, and actually most of them, even that are not in the United States, have to register at a site called clinicaltrials.org. And so, .gov, G-O-V. And so if you go on that site and then you put in multiple system atrophy, it will actually, and then you can put in like a location, you can actually scroll through the trials that are studying multiple system atrophy, and then you can also go and look through the sites that are closer to you. You know, obviously we as part of the consortium, you know, there's 5 sites part of the consortium, so New York, Boston, Vanderbilt, Mayo in Rochester, NIH, David Goldstein, and Bethesda, and then there's a few sites that are affiliated with us that are not, you know, the 5 main centers that you can be involved in trials with. So there's a couple, but that's probably the easiest if you go to clinicaltrials.gov. That's probably the easiest way to find trials that are close to you. And then the second question was, what steps do you take to donate your brain? And that's actually a good question, and actually one of our biggest challenges, because right now I don't think that we have a good repository. You know, there used to be certain groups that are organized brain donations that we learned a lot from as part of different networks. So, for example, when I was a trainee at University of Michigan, we had, as part of the Alzheimer's organization, Sid Gilman organized this big brain donation that we had a lot of brains donated. And so that's probably one of the biggest things that we need to organize for MSA.
Yeah, there are the...
We have, I'm sorry, just to comment on that. We were also using a structure that was already in place. You know, there is a national network for autism. You would think, well, this is completely unrelated, and it is unrelated, but we use the same network with the people with the beeper and the 24 hours, 7 days a week, and it's firmly established. Anybody that wants to do a brain donation, very quickly things are organized, they get all the consent forms, and whatever place in the country it occurs, we have a method, a whole national network to procure the tissues and send them to NYU. So I want to emphasize to you that that's already in place, it's working, and monthly we get other donations, and I would encourage you to know that that exists, right?
And of course that's not a subject we're comfortable dealing with, but it's very important. And there are other programs at NIH. I think if that information is not on the MSA Coalition website, perhaps we should include it. In terms of the clinical trials, I have here a question that is the opposite end of that spectrum. And the question is, can you provide any information on STEMICS, which is a stem cell clinic in the U.S., and other medical clinics? This is a very difficult question for me because my own opinion is that it is very hard for me to recommend a treatment that we have no clue whether it works. The nature of a clinical trial is, of course, to find if a medication is effective, but also to find if it's safe. And you might think, well, you know, I have MSA, how can it be worse? Well, you know, it can be worse. I mean, there's some clinical trials that unfortunately have shown that you can have so many side effects that you might be worse off. So I'm really reluctant to recommend going to these for-profit stem cell clinics But I understand full well that I'm not in your shoes. If I were in your shoes, perhaps I might have a whole different opinion. But I would appreciate the comments of other people.
I think I have a stronger opinion on that, having seen patients who have had terrible complications from what we call stem cell tourism, where I feel like this is an exploitative practice, where patients who are vulnerable are given treatments that are completely not tested, and they're often unsafe. So I would urge you to be extremely cautious about anyone who is offering you any kind of treatment for which they cannot show data, because I find many of these clinics are quite exploitative, and we have seen patients come back to our centers with debilitating complications that have made them much worse than they were. So my opinion on that is particularly strong.
I fully agree with Vikram, and I understand where Italo is coming from. He's a southern, polite gentleman. That's the way he's putting it. Now, I'm not a southern, polite gentleman. I'm a crass New Yorker. So I'm telling you, any place you go that wants to charge you for a clinical trial, run away. It's as simple as that. They are just taking your money and laughing all the way to the bank. There's no research that should charge the patient. A very different story is crowdsourcing, is what Vikram talked, what the MSA Coalition is trying to do to raise money for research. That's not charging each patient. Anybody that wants to charge you for a clinical trial, run away. It's a scam. It's as simple as that. I would even agree that patients may not show you data, because the data may not be there, but if they ask for your checking account, run away.
So there are additional questions. The first one here is there any overlap of neuroprotective research that's occurring in Alzheimer's and MSA? They give the example in this question of vitamin D levels, the correlations with developing Alzheimer's. Can some of that be useful for MSA? And the same person has extended the question to saying, is there any neuroinflammation research that's going on in the treatment of Alzheimer's or MS or Parkinson's that can be applied to MSA? And the answer to those questions is absolutely, and they should be applied, but they need to be carefully compared and contrasted. And so we need to understand how these diseases are similar. So for example, Biogen released some very interesting data for an immunotherapy against the beta amyloid protein that seemed to slow the progression of Alzheimer's. Again, an early study. We don't know if it's going to verify in a larger trial. But a good example, because if an immunotherapy against the beta amyloid protein could slow the disease down, and we get to that answer quickly for a common disease like Alzheimer's, then we have a great chance of applying a similar therapy, an antibody to alpha-synuclein, to a disease like MSA. So that's a good example. It needs to be tested. There's no guarantee, but that's a good example of crossover. General processes like neuroinflammation, absolutely, there are great potential for crossover again between the diseases, a huge amount of interest in a different type of glue cell, a microglial cell, in the field right now, and how it is perturbed in Alzheimer's disease. Could that help in MSA research? Absolutely. But I caution you that a one-size-fit-all strategy may also not work, and so that we do need to understand the biology at a deep level so that we know when to try those kinds of things and when not to.
Thanks so much, Vikram. I mean, what Vikram is mentioning, this treatment, let me add another one with the same sort of proof of concept, and is the data on a disorder called TTR amyloidosis that produces a peripheral neuropathy. And of course, these are very early results, but have given a huge boost to the whole field, because the initial clinical trial on this form of amyloidosis that destroys the peripheral nerves, depositing proteins, the mechanism that Vikram was telling you, but it is another protein in another tissue, and there was dramatic slowing of the progression. unblinded, small trial, but the results are there and they're incredibly exciting. Biogen also is another company, is thinking in the same strategy for multiple system atrophy with the same concept, which is also, I think we are all waiting anxiously and talking with them, trying to accelerate the beginning of this trial. So I have one question here. It says, my daughter is of childbearing age, and she would like to know if there's any possibility of an hereditary link for MSA. Let me answer. Is there any possibility? The short answer is we don't know. Definitely there is no, the inheritance is not what we call simple Mendelian inheritance. It's not like having black hair or blonde and then your son or daughter will be one or the other. Those are simple genetics. But genetics are much more complex than that, and that's what Vikram was talking about. Whether there are genes that increase your susceptibility to this disease, well, that's a good possibility. We don't know, and that's what we are trying to find out. So, is there any possibility that this is hereditary? Well, hereditary, not like Mendelian direct inheritance, but are there genes that make you more susceptible? Very likely, but we don't know of any yet, and we really want to find them based on the type of work that Vikram is suggesting, right?
Yeah. Going to the southern we don't have, I don't think we have very good examples where this is passed on from mothers to daughters or now. I mean, I cannot guarantee you anything. I can drop dead as I walk out the door, but that's probably as much chances as we have for it to be inheritable.
Could I just suggest, though, that if there are families, and I have now started to see an occasional family, again, very rare, where a patient will come to me and say that, my mother or my father was diagnosed with Shy-Drager disease. Very unusual for someone to walk in the door and give me that diagnosis. And often we can't track the brain, or we don't have tissue samples, so we can't confirm it. But I know that many people have contacted me online and said that, I have relatives who have dementia with Lewy bodies or Parkinson's disease, and so is it true and unrelated, or is there a relationship between the synuclein and the families? I think we don't know, and I think that the general answer here is that this is not a hereditary disease in any of the kind of conventional ways we think of that, but if there are families in which synuclein-related diseases seem to be tracking generation to generation, then please make yourself known to the research community, because we need to track such families and see if there's more we can understand about the disease from them.
Sure. I would add even one more thing to that too. Like when I was in medical school in the 18th century, Parkinson's was the example of a non-genetic disease. Let me start with that. So we all repeated, Parkinson's is not genetic. We would answer these questions very, very authoritative. No, it's not genetic. Well, there are 25 genes now we know that are responsible for Parkinson's disease, even autosomal dominant, meaning you just need one copy of that gene to get Parkinson's. Now, so...
but that's only in Venezuela, in smoke. So
what do you mean in Venezuela? Jews have increased incidence of Parkinson's. Yeah. Okay. There are several genes.
So in Parkinson, I think there's a much better evidence than in multiple system atrophy.
The difference is that Parkinson's affect 1 million people, whereas MSA affects 15,000. Right. Yeah. So Italo is right. There's no evidence. The fact that we don't have evidence now doesn't mean that we don't have to keep looking. And each time we hear, like I heard today or yesterday, families, like what Vikram is also mentioning, families that tell me, my cousin had a type of Parkinson's, the other one had dementia, somebody had multiple sclerosis. Initially, everything sounds completely unrelated. But we don't really know if it's unrelated. So each family that tells you, I have many neurological diseases in the family, I don't have an answer, but we are very curious. We said we need samples of that, and we need to look at that very seriously.
But the other challenge is that we call Shy-Drager everything. Everybody that has orthocerebral tension, a lot of physicians even call it Shy-Drager. So it's a term that is being misused. But I agree with, I think we're having this discussion, and you can see we don't agree 100%, but in the end, we do agree that we need to look into this. Obviously, it is an important issue, but I don't like for you guys to go home with a concern that, your children are going to get it, because I think it's very unlikely. Yeah, there might be a predominance genes that will make me more susceptible to have sarcoidosis, I don't know, whatever rare disease, there might be that susceptibility, but that's not the reason why I didn't have children. Let me put it that way.
Yeah, basically we're saying that if there's a genetic component, like with late onset Parkinson's disease, these are complex genetic diseases, which means there's a combinatorial, a combined effect of multiple genes potentially with environmental factors. So that's a complex genetic disease. And what that means is that there's not direct transmission from generation to generation because the likelihood of inheriting that same combination of factors is low. Yeah, there's a question.
Thank you. I appreciate it. And I want to say, this is my family. This is our second year. My wife and I come here from Alpha-synuclein, Indiana. And the one thing I want to compliment you, sir, and I'm sorry that the name of the second gentleman comes up.
Vik is easy. Everyone calls me Vik.
Dr. Vik. But I want to say that you got through the meeting like I've never been gotten through before about the possibilities, the hopes, the challenge, everything that you've been saying so far has been so spot on that I am totally impressed with you, and I don't think that'll go to your head. You've got a Quest. I went to Quest in Michigan just a few months ago, and to show you what a good spirit they had there, and this gentleman next to me across the table here will verify it. They're kind, generous, and very attentive to our needs. They were going to pay me $2,800 to do the trial. I didn't meet the requirements in the middle of the test, and they returned $1,725 to me after five or 6 days of attending there. They were obviously very astute, careful, and caring about the process that they were going through. And just unfortunately, it didn't work out for me. But I will be looking for trials because I want to be somebody that does something to help bring this to a better end. Thank you.
So we have a question here of how much sharing is done between different research centers. And this is something that we feel very strongly about as a group of collaborators that I'm sitting here with, that the field can only move forward if we all collaborate together and we share information. So right now, what we've spearheaded with Vanderbilt University and NYU is this collaborative project of the natural history of MSA, in which we have different centers that are providing their data and sharing their clinical data that comes from their patients with MSA, and we're putting that together. And the strength is really in numbers here. So what we've been able to do with this information that comes from the clinic is look at certain patients that we believe and identify those that are at highest risk of developing future MSA. So the field is coming together. The international research community are beginning to work together more strongly, and that is really thanks to the wonderful collaborators we have and the MSA Coalition that's really spearheaded that opportunity for us. The next question that came up from the audience, which is sort of on a similar line of the same project, is how much can enrolling in the global MSA registry impact research? And the answer to that is we believe that there really is a very strong reason why you should enroll in that registry for two reasons. First of all, because MSA is a sporadic disease, and like we told you, it's difficult to assemble a sizable population to do clinical trials quickly. We want to find a way in which when we do have these compounds that come up and we want to test them, that we can quickly notify you so we can get the community engaged and recruiting into trials. So that's one reason. And the second reason is what we alluded to in our presentations earlier, is that having your voice alongside that clinical information really helps us to see, are we on track? When we look at something in the clinic, is it the same as what you're experiencing at home? And second of all, what are the things that really matter to you? And those are not necessarily things that we can pull out from the clinical charts. Because when you go through a clinical chart, you can find lots of very informative clinical information, but you may be missing certain things like how quickly symptoms deteriorate, how much they impact quality of life, and that can help us as a research and clinical community come together and start focusing on the things that are important to you and your lives and what impacts you the most. So I would encourage you all to enroll in that, because I really do feel strongly that this is something where you can help shape future research.
I would just add to that, because that kind of goes along with this question, what tools would be used to show improvement in MSA symptoms in these trials, and that kind of goes along with having a large group of patients where we can ask you questions and get surveys of symptoms. Right now, one of the problems that we have is that the main way that we see benefit or look for benefit in clinical trials is simply through our exams, which are good but not perfect, as far as how we rate tremor, how we rate patients' movements, and so forth. And you don't want to wait for final things like wait until somebody passes away to decide, okay, well, this patient lived 7 years, this patient lived 3 years, oh, this treatment must be better, because one, that takes a lot of time and a lot of money, and you don't want to wait that long. So having a registry where we can, be able to build better tools to assess symptoms and signs of MSA and biomarkers to identify MSA earlier all goes along with having everybody engaged and having everybody, in a registry that we can follow.
Let me just make one comment to add to this very important thing that Amanda mentioned. The Holy Grail, the big problem or difficulty that we have is what's called outcome measures. Right? We have a treatment. This is what everybody was before alluding to. We have drug X, for example. How do we know relatively quickly whether the drug is doing something? We know that there's a progressive decline in function in people with MSA, right? Or in Parkinson's, there's a progressive decline. So we say, just to make up a number, there's 10% worsening yearly. Right? So we have drug X that it doesn't cure the disease, but maybe it reduces the fall from 10%, let's say, to 5%. Well, that's a very useful drug. But how do we find out that that happens quickly enough if we only have the clinical exam? Now, the clinical exam can be very useful, but may not be as sensitive to detect those things. So we are in a quest to find what's called outcome measures for clinical trials. One we have that we are working on, and I would encourage you to look at it, and maybe Larry Kellerman will mention that. We are looking at the retina. The retina is the back part of the eye, is a part of the nervous system, and the good thing is that you can look at the retina and take a picture. So it's non-invasive, and we have a method called OCT. It's a machine that is has much more accuracy than an MRI. You can actually almost look at the cells. We looked at people with MSA, and we saw that although they don't have symptoms in the eye, they do have a progressive reduction of one type of cell in the retina. And it looks that it's reproducible and real. So now we have a big study which is funded by the Michael J. Fox Foundation, in which we are looking at people with MSA, people with Parkinson's, and people with other similar diseases, and following them twice a year, just with a picture of the eye and an exam, a clinical exam. Anybody that wants to participate with MSA, not only we would delight it and encourage, we would actually, like you heard from this gentleman, we can even cover some of your expenses because the Michael J. Fox is paying for that. In fact, you will be paid for each picture. So any of you that thinks about the possibility, we will be more than happy to do it. That's already the first part of the study is published. It was the cover of Movement Disorders, and then we found that in pathology, and Larry will talk about that, in the actual eye of patients with MSA, we found that this reduction in a specific type of cell was really there, that what we saw in OCT was really there. Is this going to pan out to be a good way to quickly look at the effect of drugs? We hope that. We are trying to look, I cannot assure you that, but it's one way of looking at outcome measures. That, looking at MRIs in detail, like you saw today, that Dan and Vikram showed you, if we can measure these areas in the brain with a lot of accuracy, we can see if the reduction in size is slowed by the treatments we have.
Okay, thank you. I'm sorry. We are already a little bit over time, and we need to vacate the premises fairly promptly. So I've got these two questions and one other question that's come from the audience. I'm going to ask that we address these last questions that came in, and I'm going to ask the panel to please be as brief as you can in doing so. I want to at least try to give a sense of an answer to these people who post their questions, and then we are going to have to conclude the program. Okay? Thank you. So here's your last two questions if you want to. Do you want me to do this one first? Okay,
My question is for the doctor from Boston. Dr. Newman, from New York said that I think you said cynical New Yorker. Well, I'm a cynical New Englander. I'm also a Patriots fan and also aware of what goes on at BU about Chris Nowinski and donation of brains. So it's about donation of brain cells or brains. I was going to donate my brain to Chris, but now I'm provoked, Vic, by you to give it to you. Can I give my brain to both?
Yeah, absolutely. I mean, that's part of the process that we need to figure out as a group. But we need to be able to share material, okay? And so I think one of the major, in my view, one of the major challenges is how to create an integrated, for a rarer disease, how to create an integrated system so that anyone who has a good idea has access to the tissue or the cells or the genes that people have kindly donated. So in some cases, this is, for example, Horatio's group has processed samples that have then been sent to Boston to us, and it's been done on a case-by-case basis. That's been fine, but it's not good enough. It needs to be more systematically organized, and that's exactly what I think is coming through from Lucy's comment, that by creating a registry, we'll be able to access this tissue or these samples wherever they are, and have an interface that people can get to online. I think this relates to the question that I have now, so I think that's the same question, and there's a general question of, is there any relationship of MSA with Alzheimer's disease apart from just being neurodegenerative diseases? I think in some ways we've gone through those. There are very distinct proteins that misfold, but there are common themes. So I think that some things can be applied from one disease to the other, but each disease needs to be studied individually.
I know we're running out of time, but it's an important question that has to do with funding and whether North is funding this or the budget has been cut. So the importance of organizations like yours is not that you will fund the final research that will bring a cure. You provide seed money, whereas we can grow a pilot project and have enough data to submit to NIH. NIH still, by far, is the biggest funder of research. And we need NIH desperately. These are uncertain times, as you know. So any advocacy that you can do to improve funding from NIH for this type of disorders, I think will be very welcome. Thank you.
I would emphasize what Fellow just said. In addition to giving seed money, it's incredibly important any lobbying that you can do for the disease in Washington, to pressure. look, Congress, as you well know, is susceptible to pressure and lobbying. I mean, if you don't make your voice heard, there's not going to be funding in NIH for this disease. So it's very important what the coalition does, not only in terms of funding specific projects, but increasing awareness and lobbying in Washington. Lobbying in Washington is crucial to get more funding directly to MSA. I mean, wouldn't you agree?
Pam, do you want to say anything? Okay. All right. Thank you very much to our experts who have given their time here to us today. So you know we've been monitoring the live stream all day. We need to give a shout out to Perth and Brisbane, Australia. And back in the good old US of A, we've got a nice presence from in the middle of Montana. They're home. Your group's there behind you, Hadley. So, I would love to thank every individual, but there's obviously not time to do that, and I certainly wouldn't want to overlook anyone. So I just have to say thank you to everyone, from the speakers to the committee to the board members to the sponsors, the exhibitors, and all of you, the patients and families that have come together today to help us move forward together as a community. I hope that you see that there's a lot being done and there's many caring individuals here for you. You are not alone, and I hope that you walk away from this conference knowing that, and knowing that there is hope and there's people behind you. I hope that it proved to be beneficial for you. Again, I want to ask you, please complete those evaluations and get them in that box outside on the registration table on your way out. Thank you very much for coming, and email us and contact us anytime you'd like.
I think we also need to provide a big round of applause for Cindy leading the MSA Coalition. Have a great afternoon, everybody.
At the top of the exactly. More pure. Somebody like Robertson is worthwhile. I can invest it. I'll fund you. You can prove to somebody who's winning or somebody like that. No, that's great. Okay. Thank you.
And Diane wants to do a fundraiser. Next bucket. She wants to do a fundraiser. Yeah. So I'll tell her that you're the guy. I don't know what she wants to do, but I love the idea of this bicycle jersey. You know, Aaron Williams is out there. And Aaron's got a fundraiser.